Hello everyone, my name is Zobnina Natalia and today we discuss topic about fatty acid synthesis. Fatty acids are carboxylic acids that contain more than 10 carbon atoms. They have unbranched carbon skeleton and one carboxylic group in their content. In order to nature of bond between carbon atom, fatty acids are divided into saturated and unsaturated. Palmitic, stearic, lauric, and myristic acids are corresponded to saturated natural fat acids. All of them have even carbon number and one carboxylic group. Alleic, linoleic, linolenic and arachidonic acid are unsaturated natural fat acids. They have the following common signs. At first, they have even number of carbon atoms too. Also, they have one carboxylic group in their content. Another sign is cis orientation of hydrocarbon radical near each pi bond. The first pi bond in alleic, linoleic and linolenic acids locates near the 9th carbon atom. According to biochemical function, unsaturated fat acids are divided into essential and non-essential. Only alleic acid is essential, or it may be synthesized in our cells. Another unsaturated fat acids must enter together with nutrients in plant oil content. Fat acid synthesis takes place in liver, kidney, adipocytes, brain, and mammary gland. Acetyl coenzyme A, malonyl coenzyme A, and reduced coenzyme NADPH are sources for this process. Fat acid synthesis flows in 3 steps. The first is acetyl coenzyme A transport into cytosol from mitochondrion. Acetyl coenzyme A can't enter through mitochondrial membrane spontaneously. But mitochondrial membrane is permeable for substances with amino group or hydroxy group. That's why acetyl coenzyme A transforms into citric acid under citrate synthase action. It's the first reaction from Krebs cycle. Citric acid enters to cytosol and decomposes to acetyl coenzyme A again and oxaloacetate under citrate lyase action. Melanyl coenzyme A is required for fat acid radical elongation, and its form from acetyl coenzyme under biotin dependent enzyme acetyl coenzyme A carboxylase. Reaction is conjugated with ATP spent. Oxaloacetate transforms into malate under cytosolic malate dehydrogenase. After that, under Malic enzyme action, it forms pyruvate. Reduced coenzyme NADPH plus H forms as co-product in this reaction. Another source for NADPH plus H formation is direct glucose oxidation that flows at the same tissues like fat acid synthesis. Pyruvate may be returned to mitochondrion together with proton because of energy formation from ATP synthetase that locate in mitochondrion. The second step is fat acid formation. This process occurs with fat acid synthase action. It is complex multi-enzyme that locate in outer membrane layer of mitochondrion. It consists of from 2 subunits that include 7 enzymes. All enzymes are located in mirror order compared to each other. So, fat acid synthase is dimer that have X letter form in space, and it may work only as dimer, not like monomer. Fat acid synthase includes the following enzymes: acetyl transacylase, cataacyl synthase, melanyl transacylase, hydratase, enayl reductase, cataacyl reductase, and thioesterase or deacetylase. Each monomer in fat acid synthase includes acyl carrier protein. It contains residue of pantothenic acids with mercaptor group. Another mercaptor group is corresponded to residue of amino acid cysteine from cataacyl synthase subunit. Now let's describe how does fat acid molecule form. Primer acetyl residue adds to mercaptan group from amino acid cysteine under acetyl transacetylase action. Malonyl adds to acyl carrier protein under malonyl transacetylase action. After that, reaction will be continued on another part of dimer. Under keta-acyl synthase action, residue of acetic acid will be transferred at alpha position in malonic acid residue. Carboxylic group from malonic acid will be eliminated as carbon oxide. As a result, keta-acyl acyl carrier protein forms. After that, keto group will be reduced to alcohol group under keta-acyl reductase action. Coenzyme is reduced NADP. Hydroxyacyl acyl carrier protein forms as intermediate product. After that, dehydration reaction takes place. It happens under hydratase action. As a result, crotonyl acyl carrier protein will form. Then, unsaturated radical will be transformed to saturated under enoyl reductase action. Coenzyme is NADP H plus H. Name of product is butyryl acyl carrier protein. So the first saturated radical forms. After that, elongation must happen under malonyl coenzyme adding. But malonyl can be added to only acyl carrier protein. That's why butyric acid residue must be transferred or returned to mercaptan group from keta-acyl synthetase subunit. This process happens under acetyl transacetylase action. Acyl carrier protein becomes free, so next malonyl coenzyme molecule will be added under malonyl transacetylase action, and all process will be begin from the beginning. Two molecules of fat acid are formed in fat acid synthase at the same time. It happened because process flows in two opposite directions. This way helps to do fat acid synthesis more effectively. Final product of fat acid synthase action is palmitic acid. Under thioesterase action, residue of palmitic acid becomes free and enter to cytosol. Here is general equation for palmitic acid formation. As we see, 1 mole of acetyl coenzyme A and 7 mole of malonyl coenzyme A must be spent. After that, elongation and oxidation of fat acid take place. It's the third step in fat acid synthesis. Elongation of fat acids happens under elongase of fat acids. This process takes place in endoplasmatic reticulum. One molecule of malonyl coenzyme A is required for this process. Sequence in elongation reaction is the same like for fat acid synthesis, but malonyl coenzyme A plays a role like acyl carrier protein. Stearic acid forms in liver. Fat acids with carbon atom more than 18 may form in brain cell. Single unsaturated fat acid or oleic acid forms under desaturase or fat acid oxygenase action. Coenzyme for this reaction is reduced NADPH plus H. Another unsaturated fat acids may form only in plant cells. That's why another unsaturated fat acid must enter in our body together with nutrients. Fat acids formation is controlled by acetyl coenzyme A carboxylase action. Its activation is controlled by two ways. At first, acetyl coenzyme A carboxylase activity may be increased with citrate and ATP present. It means that cell accumulates enough energy for anabolytic process. Estrogens promote fat acid synthesis too. Palmitate, like final product in fat acid synthase action, inhibits acetyl coenzyme A carboxylase. Also, fat acid formation may be inhibited under another long chain fat acids that enter together with nutrients. Another mechanism for acetyl coenzyme A activation is covalent modification. Insulin promotes acetyl coenzyme A carboxylase activation. It happened because of two reasons. At first, insulin promotes translation for acetyl coenzyme A carboxylase like protein. The second, insulin impact on phosphoprotein phosphatase and transforms acetyl coenzyme A carboxylase in active state. Hormones with opposite action, like adrenaline or glucagon, inhibit acetyl coenzyme carboxylase action. It happened because of protein kinase action. As a result, acetyl coenzyme A carboxylase adds phosphate in their content and transfers into non-active state. Thank you for your attention. Wish you all the best.